In this module, we're going to talk about the biological molecules, the four classes of biological molecules that you find in all living things. And to do that, I first need to introduce you to the organic compounds, which is what these biological molecules are. They are organic molecules. And organic molecules are basically carbon-containing compounds. Of course, there are things that are inorganic that contain carbon, but these organic molecules are made up primarily of carbon and just a few other types of elements. And there is a reason why carbon is such a great base for these large organic molecules that make up us as living things. Carbon has an atomic number of 6, and it makes 4 covalent bonds, covalent bonds being the very strongest bonds. These 4 covalent bonds are made with either other atoms of carbon, atoms of oxygen, hydrogen, nitrogen, phosphorus, or sulfur. So what we're going to see are these large molecules that are based in carbon, and very often based in long chains of carbon with also covalent bonding to these other types of elements. Carbon as an atom is a relatively small size, and it can form an infinite number of structures, and these are the structures that I'm going to go through now. The simple carbon-based molecules we call the hydrocarbons because they're all carbon and hydrogen. Now in living things you don't find straight hydrocarbons. These are made up of all nonpolar covalent bonds where the electrons are being shared evenly between carbon and hydrogen. And so molecules like this, ethane or propane, you don't have to memorize the names and structures of any of these basic hydrocarbons. But these types of small hydrocarbons have no attraction to anything that are charged or polar. And so these are actually the types of molecules you find like in natural gas. They are the remnants of the breakdown of things that used to be alive. So in living organisms, we've got these hydrocarbon-based molecules with additional groups that make them polar. Well, as a living thing decomposes, what's left behind are the hydrocarbons. And it turns out that nonpolar covalent bonds, just the way that the electrons are arranged in the bond, are very high energy bonds. And so the energy that we find associated with fossil fuels is in these bonds between carbon and hydrogen. Notice that these really simple hydrocarbons can vary in chain length. We can imagine an infinite number of carbons here, each carbon covalently bound to another atom of carbon or to one of hydrogen. They can also vary in branching. So here we have a chain of 3 carbons and a 4th within the chain, but this 4th is branched from the center carbon. We have another chain of 4 carbons, but these are fundamentally different in structure. And so if you imagine a molecule with a chain of carbons, a linear chain, versus some kind of molecule with a branched chain of carbon, these two types of molecules are going to fit differently with other molecules, kind of like how a key is specific for a lock. So even though we've got basically the same general number of carbons and hydrogens, the structure is different, and so that's going to give the molecule a different function. But of course, this is still a hydrocarbon, strict hydrocarbon, not something we actually find in living molecules. We don't find butane and isobutane in living things. I said molecules, I meant living things. So we're looking at these simple hydrocarbons, but in a little bit we're going to look at these additional atoms that are added to these to make the actual true biological molecules. So far I've shown you that the skeleton, the basis for the molecule, can vary in the length, number of carbons that are in the chain, and we can also vary in whether or not there's branching within the chain of carbons. Between carbons we can also see double bonds. This is 2 atoms sharing 2 pairs of electrons rather than just 1. And here we have the same type of setup, except we've got a double bond in between these 2 carbons rather than these first 2. And again, these are different enough molecules that they have a slightly different shape. And last, these hydrocarbon base skeletons can be arranged in rings, where instead of a straight chain of carbon atoms, here we've got a ring of carbons. And check it out, we also can have these covalent double bonds between the carbons even within the ring. So again, different number of hydrogens here because of the presence of double bonds, but still carbons and hydrogens in a ring, and this has a very different structure because of the double bonds and the fewer carbons. I also want you to notice that for every carbon, there is always 1, 2, 3, 4 covalent bonds. So here I've got a carbon atom bound to 1 carbon, 2 carbon, and 2 hydrogen. Here, because carbon is sharing in a double bond with an atom of carbon, each one of these carbon atoms has only 1 hydrogen, so that there are a total of 4 within the molecule for each carbon atom. Now, like I said, we don't find strict hydrocarbons in living organisms. These are not biological molecules in that they're not actually functional as hydrocarbons. Biological molecules have the addition of functional groups, and these give chemical reactivity to a hydrocarbon, and they also give these hydrophobic, which means water-fearing, hydrocarbons a more hydrophilic, which means attracted to water, quality. So these molecules that are made up of just carbon and hydrogen, these are extremely hydrophobic. They're not attracted to anything polar, which is what water is, or anything charged. So the word hydrophobic is based on being afraid of water, but we also have to remember that not only do we have water in cells, but we also have many other molecules that are polar or charged. Regions that have carbon and hydrogen are extremely hydrophobic, and they're not attracted to anything else that is hydrophilic or to water itself. The functional groups allow these things to be hydrophilic, attracted to water and other hydrophilic substances, and they also give groups of atoms that allow for chemical reactions to happen between molecules. I'm going to describe functional groups to you, and note that we find these functional groups on thousands of different types of molecules in the cell. Wherever they're found, they behave similarly, and I'm going to describe some of that behavior right now. Some of the important functional groups, or they're called chemical groups in your text: hydroxyl group, carbonyl group, carboxyl group. In color, the group of atoms that's attached to a hydrocarbon skeleton. So for example, I have a small hydrocarbon here, and in place of one of the hydrogens at the carbon, we've got a hydroxyl group, which is an oxygen and a hydrogen. This is an extremely hydrophilic end to the molecule because of this polar covalent bond between oxygen and hydrogen. Molecules that have hydroxyl groups are called alcohols. Carbonyl group is when a oxygen is double bound to carbon, and then the fourth covalent bond is either to hydrogen or to another carbon when the carbonyl group is in the middle of a chain. When the carbonyl group is at the end of a chain of carbons, it's called an aldehyde, and when it's in the center of the chain of carbons, it's called a ketone. The carboxyl group is carbon in the chain, it's a terminal carbon at the end, double bound to oxygen, with also what looks like a hydroxyl group. But we don't call this a carbonyl plus hydroxyl, we call the entire structure a carboxyl group. This thing tends to ionize in water, just like those water molecules themselves, and they tend to lose hydrogen, the proton, in solution. It's because this oxygen here is tugging hard on electrons, because this is a polar covalent bond. This oxygen is tugging hard on electrons that are being shared with hydrogen, and the overall tug on electrons allows the electrons to stay with oxygen while the little hydrogen is released. So this thing acts as an acid in aqueous environment. It releases H+ ions. Notice that all three of these groups give a hydrophilic quality to something that's otherwise very hydrophobic. The amino group is a nitrogen attached to a carbon in a chain and two hydrogens. This, however, acts like a base in solution and tends to pick up hydrogen. So in aqueous solution, this is a positively charged group, and it actually acts as a base because it decreases the number of H plus ions in solution. Two other groups, phosphate group, which is a series of phosphate atoms, each surrounded by oxygen. Each of these were originally part of hydroxyl groups, and the hydrogens are all lost in solution. A phosphate group gives molecules a very negative charge. And of course, if something has a negative charge, it is hydrophilic. It's attracted to positive things, and it's also attracted to water or anything with that slight positive charge at one end of a pole. And then last is the methyl group. And the methyl group actually looks just like part of the hydrocarbon because it's a CH3. This is a specialized functional group that I'll only mention maybe a couple times, and just know that it's the addition to a basic hydrocarbon chain of an extra CH3. This is the methyl group, and if something has a methyl group attached to it, it's called a methylated compound. Alright, so these functional groups are clusters of atoms that are added to hydrocarbon skeletons. And you find the functional groups in all types of organic molecules. The types of organic molecules that we're going to talk about, there are 4 classes of organic molecules, are for the most part analogous to beads on a chain. So for example, one of the macromolecule classes or biological molecule classes that we're going to talk about are the proteins. A protein is a chain of beads, and the beads are individual amino acids. Carbohydrates, another class of biological molecule. A carbohydrate is a chain of beads, and the bead is a monosaccharide. So before we look at the individual molecules and the monomers that make them up, I want to talk a little bit about how they're synthesized and broken down. Imagine that we have a chain of beads here, and these beads represent any of the building blocks of the 4 classes of macromolecules. For example, your cells are constantly making proteins to do a variety of jobs in the cell. So it's sort of like, your cell is sort of like this manufacturing center of all thousands of different proteins that all have jobs to do in your cell. And in making proteins, what the cell is doing is it's stringing together beads, the beads being the amino acids. So if we were to imagine, kind of take a snapshot of building a protein in the middle of synthesis, we have this growing chain of beads, and another bead is going to be added on to this end where there is a hydroxyl group attached to a hydrocarbon chain. This unlinked bead is going to be joined right here so that a 4th bead will be on the chain, and again we'll have a free hydroxyl group to participate in a chemical reaction to add the next bead. These types of reactions, these synthesis reactions, are called dehydration reactions. Sometimes they're also called condensation reactions, but ultimately we're going to pull water out of the reaction so that you have this linkage between beads and a molecule of water has been released. Now, not only do you make proteins, but also the cells of all living things make proteins in order to do certain functions. So you know that when you eat a steak, you are eating lots of protein. You're eating the protein-rich portion of a cow, for example. The cow made lots of proteins in its cells, but it didn't make those proteins for you to eat. It made the proteins so that it could stay alive and its cells could do their jobs. You take the protein in and then you break it down into the individual monomers so that you can reuse those monomers to make your own proteins. The breakdown reactions, where we break these beads off of the chain, are called hydrolysis reactions. Hydro means water, lysis means to split. So what we end up doing is using water in the reaction to cut one of the beads off of the chain. Water is put in, a monomer is released, and we've established that same orientation of chemical groups and atoms on either side of the products of the reaction. This is a hydrolysis reaction. We've used water to split this monomer off of the chain. So what these four macromolecules that we talk about are the organic molecules or the biological molecules. We can think of, with the exception of one, the lipids, as a series of beads on a chain. Carbohydrates, one category of molecule, is made up of a chain of monosaccharides. The chain is called a polysaccharide. Proteins, the second class of biological molecules, are made up of amino acids. The beads are amino acids, and the long polymer is called a polypeptide. Nucleic acids, DNA and RNA. The monomer is called a nucleotide, and the long polymer is called a nucleic acid. There are special names for the bonds between each of these monomers as well, but we're not going to talk about those until we get into detail about each type of macromolecule. One I've left out is lipids. That's the fourth category, and it's actually the last one we'll talk about. Not truly analogous to beads on a chain or beads on a string, but another thing that makes this macromolecule or this type of molecule unique is while all of these types, the carbs, the proteins, the nucleic acids, are all very hydrophilic, these are the groups of molecules that are very hydrophobic. And they do a couple different things. Energy storage, because there's lots of hydrocarbon, but also they make up those barriers between compartments in the cell and the barrier between the cell and the extracellular environment. So what we're going to do in the next few lectures is look at each of these classes of organic molecules one by one. Before we do that, I want you guys to take a few minutes to see if you can apply what we've learned in this lecture. And first, push pause and try to fill in each white box with the name of the highlighted functional group, highlighted in yellow. I have 4 functional groups here. I have the carbonyl group, because it's in the center of the chain, it's a ketone. I have the hydroxyl group, the amino group, and the carboxyl group. Now notice that they're not ionized. You can see the carboxyl group and the amino group in 2 ways. I showed you both the unionized and the ionized form earlier. This particular picture shows them unionized, so this one hasn't lost the hydrogen to become negatively charged, and this one hasn't picked up hydrogen. But in the earlier slides, I showed you both the unionized and the ionized form, and they both mean the same functional group. Last, I want you to take a minute to write in the name of the reaction that's considered to be a synthesis reaction. Is that a hydrolysis or a dehydration reaction? And also the name of the breakdown reaction. Just take a few minutes, push pause, and remember the names of those 2 types of reactions. A synthesis reaction is a dehydration reaction, and the breakdown reaction is the hydrolysis.